Hello, and welcome to our presentation on the structure and function of RNA in cellular processes by Deja Crossman and Gabriella McBride. We aim to provide a clear understanding of RNA and why it's essential in cells. We'll start by looking at what RNA is made of and how its structure allows it to function in protein synthesis and gene regulation. You'll learn about the main types of RNA and how each contributes to cellular processes. We'll also examine a research example that highlights how RNA structure directly impacts its function, reinforcing the concept that shape determines activity. To make these ideas interactive, we'll include 2 classroom activities designed to engage you with RNA concepts. Finally, we'll tie all this together to show you why understanding RNA matters in both research and teaching concepts. RNA stands for ribonucleic acid, and it acts as the functional version of DNA's instructions. While DNA stores genetic information long term, RNA is the molecule that allows cells to actually use that information. There are several main types of RNA, each with a specific role. mRNA is messenger RNA, and it carries genetic information from the nucleus to the ribosomes where proteins are built. Without mRNA, the cell would not know which proteins to make or when to make them. Ribosomal RNA, or rRNA, forms the physical structure of ribosomes themselves. It also helps catalyze the formation of peptide bonds, which link amino acids together during protein synthesis. tRNA, transfer RNA, carries amino acids to the ribosome in the correct order, ensuring the proteins are built accurately according to the genetic code. Cells also use regulatory RNAs to control gene expressions by turning genes on and off as needed. Overall, cells rely on RNA because it allows them to respond quickly to change. RNA connects the long-term genetic information stored in DNA to the day-to-day processes that keep the cell alive.
RNA is made of repeating subunits called nucleotides. Each nucleotide has 3 main components: a ribose sugar, a phosphate group, and a nitrogenous base. The ribose sugar and phosphate group form the backbone of the RNA molecule, which gives it structure. The nitrogenous bases, adenine, uracil, cytosine, and guanine, extend outward from that backbone. One key difference between RNA and DNA is that RNA contains ribose instead of deoxyribose and uses uracil instead of thymine. This chemical composition allows RNA to remain single-stranded and flexible, which helps explain how RNA can fold into different shapes and perform different functions in the cell.
One of the most important features of RNA is its structure. Unlike DNA, RNA is single-stranded and relatively unstable. While this might seem like a disadvantage, it is actually a major benefit for the cell because RNA molecules do not last forever. Cells can rapidly adjust protein production when conditions change. Old RNA can be broken down and replaced with new RNA that better fits the cell's current needs. The cell single-stranded nature of RNA allows it to bend, fold, and loop into many different shapes. These shapes are not random. Each structure directly determines how the RNA functions within the cell. This structural flexibility is what allows RNA to perform such a wide variety of roles. Because of its structure, RNA can act as a messenger, build ribosomes, transport amino acids, regulate gene expression, and overall, RNA's lower stability is essential rather than harmful. Its temporary nature allows cells to continuously adapt and respond to their environment. Like DNA, RNA relies on base pairing, where it utilizes utilizes uracil instead of thymine, contributing to its unique structure and function.
There are several types of RNA in the cell, and each one has a specific job in protein synthesis. Messenger RNA, or mRNA, carries genetic instructions copied from DNA to the ribosome. It serves as the template that determines the order of amino acids in a protein. Transfer RNA, or tRNA, helps translate that information by bringing the correct amino acids to the ribosome. Each tRNA matches a specific codon on the mRNA, ensuring the correct amino acid is added. Ribosomal RNA, or rRNA, forms the core structure of the ribosome itself and plays a direct role in protein formation. Together, these RNA types work in coordination to ensure genetic information is accurately converted into functional proteins. In addition to these major types, cells also use other RNAs, such as microRNA and small nuclear RNA, which help regulate gene expression and RNA processing.
This study shows that the ribosome does most of its work because of RNA, not protein. The key activity, which is making peptide bonds, comes from the ribosomal RNA. Even when most of the ribosomal proteins are removed, the ribosome can still function. This tells us that the 23S rRNA is the main part doing the job. Some proteins like L16 are still important, but mostly for helping the RNA fold correctly and stay stable. They are not the ones actually causing the chemical reaction. Researchers also found that this activity survives harsh chemical treatments but stops when RNA is broken down. This is more evidence that the RNA itself is responsible for catalytic function. Because of this, the ribosome is considered the ribozymic complex, meaning RNA carries out the chemistry needed to form peptide bonds. Studies using antibiotics also show that specific regions of the 23S RNA are essential for this process. And therefore, this paper highlights the importance idea in biology. Structure determines function. The three-dimensional shape of rRNA allows it to bring molecules together and support protein synthesis. This shows that RNA is not just a messenger. It can act as an enzyme and is essential for life.
The key finding illustrated by this figure is that peptide bond formation persists even when ribosomal proteins are largely removed or disrupted. Despite conditions that strip away many protein components, the ribosome retains the ability to catalyze peptide bond formation. This demonstrates that the core catalytic activity of the ribosome does not require intact ribosomal proteins. Interpreting this result, the data provides strong evidence that ribosomal RNA is directly responsible for catalysis. Rather than functioning solely as a structural framework, rRNA actively participates in the chemical reaction that forms peptide bonds. When the integrity of the rRNA is compromised, this catalytic activity is lost, reinforcing the conclusion that RNA, not proteins, is the essential catalytic element of the ribosome. These findings support the interpretation that the ribosome functions as an RNA-based enzyme, or ribozyme, rather than a protein-driven catalyst. This represents a major conceptual shift in molecular biology, as it challenged the long-standing assumption that enzymes must be protein-based. Instead, this work demonstrated that RNA has the chemical capability to drive complex biochemical reactions. The broader significance of this study is that it fundamentally reshaped how scientists understand the role of RNA in gene expression. Ribosomal RNA was no longer viewed as a passive scaffold, but as an active participant in one of the most essential cellular processes, protein synthesis. Additionally, these findings provided strong support for evolutionary models such as the RNA world hypothesis, suggesting that early life may have relied on RNA for both information storage and catalysis before the emergence of protein enzymes. Overall, this work established ribosomal RNA as a central catalytic molecule and expanded our understanding of RNA's functional versatility.
A relay challenge is a fun activity that gets the students moving and help them understand how RNA works in protein synthesis. We set up 3 stations around the room. Station 1 being DNA to mRNA transcription, where students will draw a short DNA sequence card. Station 2 is mRNA to tRNA matching. At this station, students convert their mRNA codons into tRNA anticodons. Station 3 is amino acid assembly. Here, students will use magnetic amino acid pieces, paper cutouts, or beads to build the polypeptide chain according to their codons. This activity helps students physically move throughout the process of transcription and translation, seeing how different forms of RNA cooperate to produce a functional protein.
For this activity, students construct a simplified model of RNA to represent nucleotide composition and structure. This paper strip represents a single RNA strand made of repeating nucleotides, each consisting of a ribose sugar, a phosphate group, and a nitrogenous base. In the first exercise, students will use the model to identify RNA components and recognize that RNA is single-stranded. Because of this, the strand can fold through complementary base pairing between its bases, as such. In the second exercise, students analyze how this folding supports function. By folding the strand, students can see how RNA forms specific structures that allow it to interact with other molecules in the cell, including its role in protein synthesis. Together, these exercises will help students connect RNA composition to structure and function, reinforcing how RNA nucleic acids actively contribute to cellular processes. This presentation examined RNA as a central molecule that links genetic information to cellular function. We began by defining what RNA is and why it matters, emphasizing its role as a working copy of genetic information and its necessity for protein synthesis and gene regulation. We then explored RNA composition, highlighting how ribose sugars, phosphate groups, and nitrogenous bases form nucleotides that differ structurally from DNA. Building on this foundation, we discussed how RNA's single-stranded nature allows it to fold into complex three-dimensional structures, and how these structures directly enable function. This structure-function relationship became especially clear when examining the major RNA types, mRNA, tRNA, and rRNA, and how each performs a distinct but coordinated role in translation. We then connected theory to research by examining rRNA as a catalytic molecule, focusing on peptidyl transferase activity and evidence that the ribosome functions as a ribozyme rather than a protein-driven enzyme. Finally, the teaching activities reinforce these concepts by guiding learners through the progression from RNA composition to structure and ultimately to biological function, emphasizing why RNA is not just a messenger, but an active and versatile molecule essential to life.